Fascinating talk. Our next distinguished speaker is Professor Aaron Zorn, who is the director of CUSTOM, the Center for Organoid and Stem Cell Medicine at Cincinnati Children's Hospital. He's also the co-director of the Digestive Health Center and the co-director of the Xenopus Model Organism Database, the ZenBase. His research lab focuses on using animal models, human stem cells, and cutting-edge genomics to study birth defects. And today he will be talking to us more about the CLEAR Consortium. Aaron.
Well, I want to thank Frederick and Melanie for putting on this fantastic meeting and for giving me the opportunity to share our research with you. So I'm going to try to share my screen. Okay. Can everybody see that okay? I can't hear you. Okay, great. Thank you very much. What I want to tell you a little bit more about today is the work we've been doing in the CLEAR Consortium on animal models and in human organoids. And this will be a little bit different perhaps than some of the other talks because I really want to get into a little bit of the molecular mechanisms underlying normal tracheoesophageal development and how genetic mutations or variants can disrupt that, and we think is shedding light into the pathogenesis of tracheoesophageal birth defects. So Wendy already introduced this, and so what I want to do today is tell you about work that we've been doing in animal models and the work that my colleague Jim Wells has been done on human esophageal organoids to try to, first of all, elucidate some of the things which are really a lot of unknown aspects of how the trachea and esophagus form in the first place, and again, how genetic variants could cause tracheoesophageal birth defects, specifically esophageal atresia and TEF. So as we've already heard, and you all know, the trachea and esophagus arise from a common foregut tube in the first trimester, often before a woman will even know she's pregnant. So we believe it's during this period of fetal development when disruptions in this morphogenetic process that's separating these two tubes is really where all of the problems begin. And so we're using two animal models to study this, both the frog Xenopus and the mouse. And so typically what we see before this common foregut tube, we see it's patterned along the dorsal ventral axis into a SOX2 positive domain, which will become the esophagus, as we heard from Christoph, and an NKX 2.1 positive domain, which will form the trachea. And then this mesenchyme in between, in the span of a day or so, this mesenchyme will separate. And so our goal really is to understand the molecular and cellular pathways of normal and disrupted tracheoesophageal development. I'm going to tell you how we've been screening candidate mutations, and Wendy mentioned this from the patient genome sequencing. And really one of our long-term goals is to model this patient genotype phenotype along with their co-occurring anomalies, and I'll tell you more about that. And what we learn about the normal development of the trachea and esophagus in animals has really been critical for informing approaches to direct pluripotent stem cells into trachea and esophageal tissue. I'm also going to take the liberty of telling you about some of the work from my close colleague, Jim Wells. Jim Wells and I have a joint lab. We've had so for over 20 years. And what Jim has done is using developmental paradigms, things that we've discovered from animal models, created ways that he can take iPSCs and make esophageal tissue. And it's very exciting. He can take these iPSCs that Christoph described, and he can independently induce, by mimicking in vitro the growth factor cocktails that occur during normal development, and make the endoderm epithelium, the mesoderm, and the enteric neurons, mix them all together and make these really complex esophageal organoids. Actually, he can make now, his group has been able to make organoids of all major organs of the aerodigestive tract. I'm just talking really up here today about the esophagus. We grow these in vitro and then transfer them to an air-liquid interface where they develop a sort of more typical stratified epithelium with mesenchyme. We get the expected keratin expression, smooth muscle will develop, and all of the sort of typical markers. And these tissues really look like sort of mid-gestation fetal epithelial esophageal tissue. we're using these now, really taking the same kinds of approaches that Christoph described to model esophageal atresia mutations, and then trying to understand the impact of those. And then ultimately, I believe you heard earlier today about approaches to generate tissue for replacement. And so this is something that he's made a lot more progress in the intestine, but he's pursuing for esophageal tissue for replacement therapy as well, and in preclinical animal models. Okay, so what I want to do today is spend a little bit of time telling you a bit more about what we know about normal tracheoesophageal morphogenesis from animal models. I will follow up on what Wendy mentioned in validation of SMAD6 as an esophageal risk gene, and show you the data we have in animal models and organoids. And then finally, I'm going to finish talking about some recent work that we just published in collaboration with Wendy and Yufang Shen, looking at a class of variants that she found in the complex patients that really suggests perhaps a conserved endosomopathy pathway that might really explain these sort of complex, non-isolated cases, and I'll explain what I mean a bit more there. So really, a lot of what we know about early tracheal esophagus development starts from animal models. So here's an example in the mouse embryo. So this is a mouse embryo, E9.5, embryonic day 9.5, equivalent to about a 30-day human embryo. And this is a confocal image showing you the gut tube in yellow. And the first indication of this common tube being patterned along the dorsal ventral axis is this ventral expression of the transcription factor NKX2.1, as Christoph was mentioning. And if we section through the embryo, it looks like this. There's really no histodifferentiation at this point. It's a simple columnar epithelial. SOX2 originally is expressed throughout the foregut tube, and then it gets downregulated in this ventral domain, which will become the lung and the trachea, and it's maintained in the esophageal domain. And FOXF1 marks these the mesoderm, and at this point, again, there's no muscle differentiation, it's just mesoderm. We and others over the last decade have really elucidated this regulatory network that Christoph was alluding to, and really the take home is that Wnt and BMPs are expressed in this mesoderm. And I should mention that this mesoderm is the same mesoderm. It's the same pool of mesoderm that also contributes to the outflow tract and portions of the atria. So it's referred to as the cardiopulmonary mesoderm, and that'll be important when I return later for our discussions on SMAD6. So it's really this gradient, as Christoph was mentioning, of Wnt and BMP, and BMPs repress SOX2 expression and in the ventral domain. And there's also this mutual repression between SOX2 and NKX. And so this is why quite commonly, if there's a disruption in any one of these pathways, this is a sort of a binary cell fate decision between cells adopting an esophageal epithelium or a respiratory epithelium fate. And so what's become very clear from single-cell genomics of human embryos and organoid work is that the molecular mechanisms of really all of these animal and humans are highly conserved. What's been less clear is really how you go from this patterned epithelium, and in the case of animals, in 2 days, really this separates into two tubes that have this stereotypical exquisite morphology. And very recently, we've been able to elucidate a lot of the cell biology that underlies this process. And so I'm summarizing that here. So initially, the foregut gets patterned. These are cross sections of the foregut here along the dorsal ventral axis, as we talked about. And then there's really at this boundary between the SOX2 and the NKX2-1 domain, there are cells actually which transiently co-express both transcription factors. And it's really at this point where there's a condensation of the surrounding mesoderm, and it tends to push these epithelial together, and then they touch. And what I'm going to spend a fair bit of time telling you about is that we've discovered this really previously undescribed epithelial remodeling. What happens is when there's this fusion of the epithelium, and then there's a resolution of this transient epithelial septum, and the mesoderm invades. It separates these two tubes, and then, of course, all of these tissues, both the epithelium and the surrounding mesenchyme, undergo differentiation. And what we've been able to show through work in Xenopus is that if we remove it with microsurgery, the mesoderm from one side of the embryo and let them develop, that it doesn't form properly. And conceptually, if any of these sort of morphogenetic processes get disrupted, this could result in EATEF. If this process paused briefly, you would end up getting a short TEF and a short gap. But if it was a prolonged period where this didn't happen, or it didn't happen at all, you would essentially get a single tube that never separated. So let me tell you a little bit about really what I'm talking about here in this epithelial remodeling. And so these are confocal images of early embryos, in this case Xenopus. We're looking now at two different sections along the more anterior and posterior, and this is the foregut epithelium. It's a pseudostratified columnar epithelium. It's apical basal polarized. In red here is the apical membrane marker. You see that this is this mesenchyme bulging, squeezing the two tubes together. They touch, and they actually downregulate the apical marker. They upregulate cell adhesion proteins like E-cadherin, and they fuse, and they form this transient epithelial septa. Now I'm just showing you a highlight here. This would be this forming esophagus. The forming trachea would be down here. And I'm going to show you this movie of how then this epithelial septum becomes resolved. This isn't a time-lapse movie. I wish it was. We're working on that. Rather, this is a confocal stack going from the region of the embryo where this epithelium has not separated to the place where it does separate. And what happens is the basement membrane breaks down. Cadherin, the cell adhesion protein, is sort of downregulated. Right here, the cells round up and they separate, and these mesenchymal cells invade to then separate the two tubes. And so we've really started to understand this at a single cell level resolution. And as far as we can tell, the same sort of thing is happening in human embryos. If you look at some of the Carnegie sections carefully, you can see that in fact, even in human embryos, we can catch this transient epithelial septa. Okay, so what I want to do now is, using that information that we have elucidated from animal models, try to think about how can we can validate some of these risk genes, and I'm going to focus first on SMAD6. So as Wendy mentioned to you, she and Yufeng have identified over 200 protein-altering heterozygous de novo mutations. The challenge, of course, is to figure out which ones are causative. And so in collaboration with Yufeng, we use data mining of animal model databases and single-cell genomics to find genes which are expressed at the right time and right place from when this morphogenesis is occurring, think about their function, and of course, if the mutation is going to cause a deficit. And then we test the genes, candidate risk genes in Xenopus. This is a high throughput system, as I'll show you. And then finally, once we have some good candidates that we have evidence from the frog system, we would move to the more labor-intensive human organoids. And I'm going to start first with SMAD6. Because we had two patients that had variants in SMAD6, and Wendy described this to you already. I won't belabor it, but these patients had esophageal atresia with tracheoesophageal fistula, but they also had co-occurring cardiovascular or renal deficits, and they had these potentially or predicted to be likely protein-damaging mutations. So what is SMAD6? Well, SMAD6 is an intracellular protein in the bone morphogenetic or BMP signaling pathway. So as many of you know, the BMP pathway controls many aspects of development, homeostasis, and is disrupted in many different settings in disease. SMAD6 is a protein which essentially restrains the intracellular signaling. So normally, of course, BMPs bind a ligand at the receptor. They transduce through these SMADs, they get phosphorylated, they go to the nucleus and they turn on genes. Well, what SMAD6 does is it restrains this activity. And SMAD6 has been mutated in mice. It results in embryonic lethality and with severe cardiovascular deficits. And the trachea and esophagus haven't really been looked at. Normally, as you all know, these EATEF in a mouse model anyways might not be noticed until the pups are born and they have trouble feeding and breathing. So we had a look in animals, and this is the expression of SMAD6 in embryos. It's expressed in the heart. This is also the heart, and this ventral mesoderm, as I mentioned, is also the source of BMP. So SMAD6 is typically expressed both in the BMP expressing cells and in the BMP responding cells. So here is an embryo right at the time when it's separating the esophagus and trachea, and you see very high SMAD6 expression in the trachea, in the septal region, and in the surrounding mesenchyme. And of course, just to remind you, so the prediction would be then that if you had a mutation in SMAD6, you might have elevated BMP signaling, and therefore you would have reduced or disrupted esophageal development. And so that really was the prediction. So we first turned to the frog model, and I want to take a brief aside and just sort of explain to you why on earth will we use frog models when you have mice and many other approaches. So I'm just going to explain a little bit, introduce this frog model. So these are aquatic frogs. We can have a colony of them here with 1,000 upstairs. These embryos, we can generate embryos, thousands of embryos on a daily basis. Here they are. Here they divide very rapidly, and so we can get thousands of sibling embryos. And so this is very helpful considering this genetic background. So we can manipulate or do experiments on some of these embryos, and then we're really comparing sibling embryos to reduce this genetic variation. The embryos are very robust and large. This is the size of a frog embryo, right around the stage of a mouse embryo of equivalent. By simple microinjection, we can use CRISPR-Cas9 or various other transgenic approaches to manipulate genes. And one of the interesting things, of course, is that these are externally developing, so you can watch the development. It's not inside the mother. They're not reliant on a placenta, and so what can happen is these are sibling embryos, and this is actually alive. It doesn't have a heart, it doesn't have a head or a spinal cord, and it'll be alive until these embryos start to feed. And so normally in mice or human, if you had a severe mutation, say in the heart or in the placenta, the embryo dies very early before organ formation, and you don't have any material in an experimental setting, in the case of mouse, to study. So in this case, we can get these embryos to the point where we can then analyze them, even if they have severe deficits. By 3 days, we have transparent embryos. Here's the eye, the beating heart. This is the digestive system. The trachea and esophagus are in here, the developing kidney. I think if you squint, you can even see the blood vessels, blood cells moving through these major vessels. And so this really gives us an opportunity to watch in real time the development of these tissues. Okay, so this is the reasons why we start with this system first when we want to test screen genes. And so in my lab, the postdoctoral fellow, Nicole Edwards, has developed this rapid F0 CRISPR-Cas9 genome editing. So she designs guide RNAs to cause indel or loss of function mutations in the frog embryo. And 3 days later, she can then use confocal imaging to then analyze the separation of the trachea and esophagus. And in this case, what she did is she made a mutation analogous about the conserved place. Not exactly the same amino acids, but this would be the conserved residue that this one patient mutation had. And the idea here is she's introducing indel mutations, which will be frame shifts or changes in amino acids. And when she does this, here's the control, separates properly. SOX2 is the positive control. This is a germline mutation. They never separate. The esophagus doesn't form, and they have a single tube. In our F0 CRISPR-Cas9, you can see the same sort of a phenotype. And now SMAD6, they have this persistent septum. And an interesting thing here is you see at the septor region, we have increased NKX2-1 expression, which might be consistent with elevated BMP signaling. With this in hand, we then moved to the Jim Wellis's group to the human esophageal organoids. And I just want to remind you, and this is sort of the proof of principle. So in Jim's lab, we've established this system, and this is the same sort of system that a number of groups are using, as Christoph described, where you can mimic this embryonic development, make foregut, and then you can direct them into esophagus or respiratory. And here's what these look like. Here's the esophageal organoid with SOX2, the NKX2-1 positive respiratory organoid. If you let these develop a little further and you look carefully, you can see that SOX2 and NKX And as a proof of principle, if they use CRISPR-Cas9 to knock down SOX2 expression, you see here you get reduced SOX and you have the ectopic NKX2-1, and that's to be expected because of this mutual repression that Christoph and I described to you. So using this system then, an MSTP, MD PhD graduate student wanted to explore the role of SMAD6. And so in the first instance, she's making a rather large loss of function mutation. We want to avoid trying to look at these sort of subtle minor haploinsufficiency. We just really want to understand, is SMAD6 involved in this process? So she in the first instance, so she makes this mutation and she differentiates both the esophageal endoderm or the esophageal mesoderm. And I'm going to show you just some of the data. This is being submitted for publication now. In the SOX6 mutation, in the SMAD6 mutation, you see that they're really deficient in their ability to make or turn on SOX2 positive esophageal progenitors. They have some ectopic NKX expression. And so this is consistent then with these cells sort of experiencing increased BMP signaling because, as I've already told you, and Christoph told you, that BMP suppresses SOX2 expression. So this would be consistent with elevated BMP signaling. And indeed, when she does RNA-seq and she looks at these carefully, and she looks at a number of well-known BMP target genes. So all of these are genes which are known to be turned on in response to BMP signaling. And in the SMAD6 mutants, these are all elevated. So indeed, the SMAD6 mutant cells, under the same differentiation conditions, have a elevated BMP activity. I don't have time to show you all the data, but she's done similar sorts of things in the mesoderm. And one of the things that she finds, again, here's the elevated BMP signaling. But as a consequence of that elevated BMP signaling, she sees that now the mesoderm is adopting more of a cardiac fate, these cardiac markers, and indeed even some more of posterior mesoderm fate. So really the idea is, what we're thinking now is that these SMAD6 mutations is going to cause elevated BMP signaling in the mesoderm, and that's really going to change the allocation of that mesoderm to how much of it is going to be in the mesoderm that's surrounding the esophagus versus mesoderm that's going to contribute to the cardiac. And that really, we think, our hypothesis could explain both the tracheoesophageal, or the EATEF, as well as the deficits in the cardiovascular system, and indeed the kidney, because the kidney is also deriving from that early intermediate mesoderm as a result of BMP signaling. Okay. So this is sort of an example of how we think using these multiple approaches, animal models in vivo and in vitro human organoids, have really implicated SMAD6 as a risk gene for esophageal atresia and can explain some of the comorbidities and co-occurring anomalies that these patients have. What I want to do now, just in this last part, is return to how we're using the FROG system as a high throughput screening platform for studying these variants. So Wendy mentioned, it's quite a daunting challenge when you have 256 de novo variants and you're trying to figure out which ones are causative. She mentioned that the work of Yufeng really came up, though, with this intriguing observation that the complex or non-isolated variants from these patients, the non-isolated cases, really were enriched in variants in genes that were involved in endocytosis and intracellular transport. And this was very intriguing to us because we had already been thinking that this was reminiscent of a process known as endosome trafficking. And I'm going to explain what this is in a moment. But what I'm showing you here are in green are these genes for which we had patient variants. And this is a protein-protein, this is an interaction map from experimentally validated protein-protein interactions in the literature from the dbString database. And we've seeded this with core components of this endosome pathway. And you see they really form this network. And so this was the first indication that maybe what we were seeing here is a pathway, a cellular pathway that might be disrupted in many EATEF patients. So what's endosome trafficking? So endosome-mediated trafficking is something that all cells have. It's driven by small GTPases, and really it's involved in moving components of cells and cell adhesion proteins from different compartments of the cell. So for example, Dynamin 2 would be involved in endocytosis, removing part of a membrane as well as the associated proteins, and then translocating that into early endosomes. And those proteins are then either degraded or moved to another component of the cell. And so really what we were thinking about is this cellular process could explain both the epithelial remodeling that we see at this fission step, as well as this fusion step, where you have to sort of, you're remodeling the epithelium, you're moving cell adhesion proteins and polarity proteins from one part of the cell to another. And so our hypothesis was that this pathway could indeed perhaps explain some of these. So Nicole went back to her screening, and she, in this case, was helped with a colleague of mine, a neonatologist, Alan Kenny, and he has a research program with us. And so here are the patient variants. Again, you see that we found that 13 out of 18 of these endosome variants. So again, we're mutating these. In this case, these are loss of function mutation. We just want to see if these proteins are involved. We see a failure to separate. In many cases, the esophagus is occluded, and it will degrade. And we also looked at these core components of the endosome pathway, these small GTPases, Dynamin 2, Rab5, and Rab11 for these recycling endosomes. These are the Rab that can move vesicles from one component to the other. And they all resulted in variably penetrant TEFs. Okay, and so Nicole wanted to dig into the cellular mechanisms a little bit more. And these are now confocal images looking at cadherin expression. So here's a control. It just separated the esophagus and a trachea. And in all of these, when we mutate these core endosome proteins, we see that the septa doesn't separate. We see these blistering. Eventually, these will reopen up again, and you have this persistent cleft, and sometimes the esophagus itself is occluded. The earliest deficit that she could see was really at the point where you form this transient septa. Normally, it's really a very stereotypical bilayer, and what she was seeing is these epithelial cells were really piling up. They fused okay, and this is really where the first place where we saw the deficit. I don't have time to go into all the details, but this was really indicating to her that there was some sort of disruption in cell polarity. And so that made her think about this cell polarity complex known as Wenger-Seltzer. So these were first described in Drosophila. They control the polarity of epithelial. They're atypical cell adhesion proteins, and they're typically associated with the apical surface. And indeed, we had one mutation, missense mutation, in our cohort that had a variant in Seltzer2. I'm skipping a lot of data here, but what Nicole was able to show, this is a high power image of this septa region. And in fact, she sees that these Rab11 vesicles are co-localized with Wenger2. So the concept here is that Rab11 is moving this vesicles containing this adhesion polarity protein Wenger2 right to this fusion point, right down the midline of this epithelial septa. And if we mutate these various endosome pathway components, either endocytosis, early endosomes, recycling endosomes, we don't properly localize Wenger there. And indeed, if we mutate Wenger itself, we phenocopy. And you see you get this pileup of cells, the blistering in the septa. And if we mutate Seltzer, which was one of our patient variants, as well as ITSN1, which is a small GTPase GEF, we can phenocopy. So really, I sort of skimmed over a lot of data that we don't have time to get into. But what Nicole showed was that during normal development, it seems that endosome recycling is important to drive this epithelial remodeling. And it's critical for translocating this polarity adhesion protein VENGL right at this transient septa. And that seems to be critical for then ultimately the downregulation of E-cadherin cell adhesion protein, the separation of this, and then the allowing these mesenchyme to invade. And when we have mutants, at least in the animal model, where we mutate these same genes that the patients had variants in, we see that this separation doesn't occur properly. VENGL is not localized. Cadherin is not properly downregulated. And we see that there's this blistering and ultimately a persistent septa, and sometimes then ultimately a degradation of the esophagus. What we're pretty intrigued about is the fact, returning to this observation, that this was really in our complex patients. And so what we're starting to think about now, as Wendy already mentioned, that really about a high proportion, 50% of these EA patients, as you know, have deficits in other organs. And based on developmental biology, we know that the heart, kidney, particularly the anal rectal malformations, the separation of the hindgut into the urogenital system, and the bladder and the rectum, that really undergoes the same sort of morphogenic process that the foregut does. And so our idea is that this may really represent a variably penetrant condition, maybe what we're thinking about of endosomopathies, similar to ciliopathies, which might explain many of the different deficits that these patients have. Okay, so really, what I hoped I've shown you is that with this very productive collaboration with Wendy's team and many of you here on the call, clinicians around the world, human geneticists, developmental and stem cell biologists, really bringing a number of different approaches to bear, patient genotype phenotype registry, whole patient genome sequencing, genomics, organoids, and animal models, we've been able to start to reveal the developmental genetics of EATEF. I think we have pretty strong evidence now that SMAD6 is a risk gene, and it can really explain variably penetrance in esophageal atresia, as well as congenital heart deficits. And we're starting to elucidate the mechanism that really looks like elevated BMP signaling, and perhaps as a result, a misallocation of cells between different compartments during early organogenesis. I've shown you that we've defined that endosome trafficking is a critical component driving this epithelial morphogenesis that separates the foregut and trachea and esophagus, and that dysregulation of this pathway can cause EATEF, enlarged laryngeal clefts in these animal models. And we're starting to think that this may account for endosomopathies, and this may be really an underlying mechanism for many complex EATEF cases. And with that, really want to thank an incredibly fruitful collaboration with Wendy, Yufeng, and my close collaborator and research partner, Jim Wells, and of course, the people in the lab. Nicole Edwards in my lab has been really driving this, and I'm happy to say she will be starting her own lab at the University of Michigan shortly. So with that, I would like to stop sharing and take any questions. Thank you for your attention.
Thank you, Aaron. The floor is now open for questions.
I know it's really different from what you're used to. So even if there's something you didn't understand, please go ahead. I'm happy to clarify.
Thank you very much, Aaron. I'm a pediatric surgeon. It's much more difficult than the other audience. But your different screens you showed, it's not a very important question, but can we put some timetable? I mean, the persistent communication occur before the atresia or at the same time. And the secondary question in the same topic, have we some hypothesis like we asked to Paulo and Katan? Can we explain why there are different anatomical form of this malformation? It's the quantity of gene suppressed or multiple gene association. why we have types with fistula and without fistula. But
Yeah. So obviously that you're absolutely right. That is the critical question, and I think we have more. We have a better understanding of a mechanistic hypothesis now. We don't have an answer. One of the things that we're working very hard on is live imaging, where we can microdissect from the mouse the foregut prior to separation, and we can culture it for 48 hours, and we use transgenic animals where either the trachea or esophagus are fluorescent, and we've had some success, and we're starting to model to image it. So that will really allow us to answer what you're asking. But I can tell you what we think so far from the analysis we've done. What I think is it depends how long during development and at what time during development this separation process is interrupted. Okay, so it's a progressive, it occurs from the carina where the lung buds are first coming up, and then it moves towards the larynx over about a 24 to 48 hour period in mice, for example. And so remember, these patients, of course, they're heterozygous, and so they have a barely penetrant phenotype. It's not like the severe loss of function that I've shown you in the animal models. And it's polygenic. So what we hypothesize at this point is that development will proceed, and then at some point it will stall. And if it stalls for a long period of time, that would end up in a long gap. If it stalls for a brief period of time, it would end up in a short gap. If it stalls early, it will end up in more of a distal TEF. If it stalled later, it would be more of a proximal. That's if there was a stall in this separation deficit. It's also clear that if the mutations result in a early misallocation in the number of esophageal progenitors versus the number of tracheal progenitors, that can have a different effect. What can happen is you get the single tube, and let's say you have only 20% of the esophageal progenitors, it'll try to constrict up at the top of the tube. And we think in that case, that's really where, I think that's actually more common. And that may result in then the esophagus just not forming properly. And so you'll have a trachea. The trachea, as many of you know, of course, your patients will have tracheomalacia. That would be a prediction from this model. You'd have a very small atretic esophageal portion, but you'd have a larger, essentially floppier trachea because you'd have a much larger trachealis muscle region. So those are the predictions from what we see on the cell biology, and now we need to do the live imaging to test them.
Thank you very much.
Frederic speaking. Thank you so much for this fantastic talk, Aaron. I have three questions if I can ask you. The first is related to SMAD6, the novel mutation. You showed very convincing data about its functional and pathogenesis pathway, but my question is, if I understand well what Wendy showed, and you also, this mutation were also only seen in 2 patients, a long list of other mutation in many other patients. So my first question is, why SMAD6 and not the other gene, and how do you think it can be generalized to patients without this lot of functional or mutation? I can't remember what type of anomaly.
Right. Well, so we think, not the SMAD6 mutations, I believe, would be the sort of case where there's a misallocation of these early progenitors. There would be too much BMP signaling, and you don't make quite enough esophagus, and you might make more sort of cardiac, and it's inappropriate. Remember, the patients, of course, they have this missense mutation, and they're heterozygous. So what we would expect is that, because of other genetic background, we would think that normally if it's polygenic, the patients might need to have multiple variants. So let's say the patient, SMAD6 patients had this particular variant. Their SMAD6 works kind of okay, and they have one normal copy. If they have some other variant, then it becomes a developmental bottleneck, and you have this stall in the process. What we're doing now to address this is going back and making the patient variants and trying to see if we can detect subtle changes in BMP signaling or cell fate allocation when we have exactly the patient mutation and we're comparing to an isogenic control. And so I think that will give us a more satisfactory answer to your question.
Okay, thank you. And my second one is about the endosomopathy theory, or what you showed about that. It's very exciting theories, and my question is very basic. I mean, I'm not a surgeon, but a physician and a pediatrician. So, are there genes regulating this endosome functioning? Probably yes. And in the table Wendy showed us at the previous talk, were there any of these genes mutated or deleted, or
yes,
in the long list of genes where mutation or deletion, loss of function?
Absolutely, and I'm sorry that probably went a bit fast. So based on her and Yufeng analysis, we found variants in 40 different genes, about 40 different genes in our patients. And that was really from patients that had these complex cases. They had multi-organ system deficits. So that's how we identified this pathway in the first place, is that we saw an enrichment in these genes in the patient cohort. And then what I showed you is how we tested the hypothesis that those genes might interact in this concerted cell biology pathway, which is endosome trafficking. And so we really do think that this could be, at least for a subset of complex EA patients, something that we need to look at more carefully.
Okay. My last question is about more treatment or perspective treatment. You mentioned in one of your first line that organoid can mimic all the levels of normal, the vagal mucosa, mesoderm, endoderm, and the nerves too. And it could be a target for replacement for treatment. We had a very nice talk this morning about bioengineering. So what would you expect it as a clinical development of such approach? Or what are the technical problems you have to solve before?
So that's a great question. Obviously, that would be the holy grail. I would say that the tissue that Jim Wells's group can make with the smooth muscle, the epithelium, the enteric neurons, it's sort of really at the level of mid to late fetal gestation tissue, if you compare it to humans. So maturation of the tissue, further maturation of the tissue is one point. The other point, obviously, would be to take it from sheets of tissue to some kind of a scaffold that would be useful for surgical perspective. What I can tell you is that my surgical colleague here, Michael Helmrath, who is a intestinal transplant surgeon, I'll tell you the example we have of the intestinal organoids so far. So what Michael Helmrath has shown in rat models is that if you have rat models where you damage the intestinal epithelium with detergent, or you could be modeling IBD or something like that, and then you take intestinal organoids, you shear them up into pieces, and then you can actually deliver them endoscopically into the rat, and in a matrix which is light activated. The light can then you put them into the gut endoscopically, and then you activate the light with the endoscope, and that hardens the material and it sticks. And what he shows is that the human cells can basically completely repopulate the damaged rat epithelium. The critical point here is that if you try only epithelial alone transplants, they don't work. You need the mesenchyme, and the mesenchyme is critical for engraftment into the damaged host. So by analogy, the first applications that we're thinking about really are probably not esophageal atresia repair. It's probably more like esophageal damage that we may see in a pediatric setting. You know, the child has drinking something, you know, that's damaged the epithelium. Yeah, caustic. And so that's really the first application that we're thinking about. And we're in the process, my colleague, Michael Helmrath's in the process of trying to develop an animal model for preclinical testing.
Thank you.
Aaron, do you think this could be applicable in achalasia patients too?
It could. I mean, it's so early to tell. You know, I think I don't want to speculate, but I think we're heading in that direction. You know, we're also even thinking about motility issues because now we have the enteric neurons. Yeah.
If there are no more questions, and I can't see any more in the, Cornelia, you raised your hand.
I raised my hand. I thank you to everyone for all the excellent presentations. It feels like we have pieces of the jigsaw that we're putting together, and it's just beautiful. I don't know whether Aaron would be the best person to reply to this question, but I was also thinking, post small bowel transplant, there is motility restored. Have you studied at all, or do you know of anyone who has studied that and how the enteric nervous system repopulates and connects with the transplanted organ?
I will leave that to one of the other participants, since I really don't have any knowledge about that. I mean, I don't know.
I'm sorry. It's just
yeah, no, it's a good question.
As everybody was talking, thank you.
Yeah, it's a good question, but I don't know. One thing what I would say is that, you know, conceptually, I think, as I've learned from all of you, that you know, these bowel transplants don't always work as well as the endogenous esophageal tissue. So I do think that having the right smooth muscle, the right enteric neurons, with the right epithelium, if we can get it to work, hopefully would produce better results.
Thank you. I would agree. Thank you.
And I guess, Aaron, following on, you could possibly apply it in the Hirschsprung sphere too, at the bottom end.
Yeah, so we're definitely looking at that. We have a bank of iPSCs. We, Michael Helmrath's team and Jim Wells, a bank of iPSCs from Hirschsprung patients of known and unknown etiology, and we're doing exactly that.
I can't see any more raised hands. Fascinating talk, all three of them. Thank you to all the speakers, Wendy, Christoph, and Aaron.
Thank you.
Thank you. We have just